Hello, everyone. Can we start, guys? I hope this is Bio-M 08, and it's the master's. Good. So I know in a different room, like I normally do. So my name is Eugenio Sanchez Moran. So I always start saying where I'm coming from. So during my PhD in Madrid, where I'm original, I decided to do something that only one person in the world could do, and that person was here in Birmingham, and now he's retired, and I'm in his office with his position, so it worked quite well. And this is something that I suppose you have been seeing already with all the different lectures that they have been teaching what they are actually doing researching. So I normally ask this, and it's quite fair in the first day because I don't know any of you, and I will never remember what you answer. Who here is interested in plant genetics? Yay, there's some people. Normally we don't have anyone, so it's quite good. So yeah, I hope this is a kind of review of what you know, but from the perspective of plants. And for the people that they are no interest in plants, oh God, we are losing one projector. Well, hopefully it comes back. If not, I use this one. Can you say this one? No, I hate this room. You can move if you want to. Are you okay with that? So I don't know. Yeah, it's just overheated or something like that. Are you okay with that from there? Yeah? Okay. So basically, yeah, hopefully all of you that might not be interested in genetics, this is actually helping you to understand from another perspective all the different techniques that they are used in any organism, okay? As you will see, I work in plant genetics, so I was trying to sell it out, okay? So there will be some bits where I will remind that actually it won't have a cyst, no, it's coming back, without actually someone finding out that problem in plants. So we got 4 lectures and we got 1 workshop that is around 2 to 3 hours, the workshop. So I will let you later, just in between the 2 lectures today, I will let you know that you need to prepare for that. And it's just basically read 1 article, okay, each. So it's not too bad. Hopefully, we will have a very good discussion during the workshop with all of you. And I will talk more about later on. So today, we are going to divide the 2 hours in 2 different things. I always like to start with a history of genetics, basically, a very broad way of bringing you to all the genetics that we know, and actually emphasize where plant genetics has been important and why it's necessary. I will continue just with different mother organisms that we use in plants. As you know, in humans, you can use cell culture, you can use other mammalian systems that you could use as a model. And in plants, we are lucky we have also these kind of models that we could use. And these days, actually, we are passing from well-known model organisms like Arabidopsis to more complex like proper crops, so wheat and maize. The second part of today, the second lecture, it will be genes and mutants. And I will talk more about that and how to do mutations and how genetic analysis actually depends on that, okay? And especially I will focus how to generate mutants in plants. That is very similar with some differences and different techniques for other organisms. So I always ask the same now. Do you want me to go through, and when we are finished, or do you want to make a break? Do you want a break? Yeah? Good. So that's better for me too, so I can talk more. So basically, this is just to remind you that, in a way, human civilization has been doing kind of genetics since the beginning, okay? Since they decided a crop was a crop or a plant that they were interested was very good to grow in the next generation, so they could have the same kind of quality of the fruit or the same amount of grains, or as how good or easy was to collect. From that point of view, human civilization has brought agriculture and actually the beginning of genetics. One of the key things that plant breeders do these days are selecting. And this is what has happened since the beginning of agriculture. Selection of crops, basically. The other part that plant breeders are doing these days and for the last 2 centuries have been hybridization. So suddenly you cross between one plant and another because you want to pass one of the characters from one to another. So these are key words in plant breeding and key things actually for knowing what genetics is, okay? And I always bring this because you know Mendel, yeah? What did he do? Anyone can just summarize quickly. Yeah?
He crossed genes and he discovered that certain traits were inherited very easily. So whether these were real smooth, yellow or green, he found that there were direct patterns between whether the parent was yellow or not and whether the offspring was yellow or not.
So the key word there is cross, okay? And this is the key thing in genetics. You need 2 things to actually do this cross. Variability, so different characters that they have different phenotypes. In this case, the peas were smooth or rough, or they were yellow, or they were green. And the same that you will need for any genetic analysis, okay? And then cross. You have to cross them to be able to see what happened, how they segregate. And basically, this is the beginning of genetics, okay? Genetics as a science, the first thing it does is actually be able to use hybridization or crosses to be able to analyze how the genetics are inherited. And this is basically what Mendel kind of discovered, really. Well, kind of not, but really discovered, although he was not too sure, as we will see in a second. So, quite important, he published his results and actually did the right interpretation of them. He was lucky because he used characters that they were independent, so the laws work quite nicely. Some people have analyzed statistically his results and they are too perfect, so he might have adjusted a little. But in a way, he actually had the right answer, so that was quite good. But it took 35 years to be rediscovered and actually being recognized by Correns and De Vries that they were working in animal cells, really. Well, Correns actually was working in plant cells too. But yeah, it took all that time to actually be discovered. Also, there are rumors, still it's not completely proved. It's in the library from, in the house of Charles Darwin's. There is a copy of this article in the library, but no one knows if he was there when Darwin was there. So we are not too sure about that. But it probably that Darwin had a copy of the manuscript but never read it. And the main reason is because it was plants. Okay? And this is something that still happens this day. If you go and talk with people working in cancer research, if the moment they see plant in the title, they will never read it. Even if there are proteins or enzymes or pathways that they actually relate to what they do. They normally just ignore it, okay? And this is something that hopefully in the future you don't do that, okay? Because you might find really important things just because plants are easy in a way to manipulate. So basically he did around 10,000 crosses. So if you think about crosses, and this is something that I will bring out in the last lecture too, is about manual work. You have to take pollen from one plant and put it in the female part of another one. So it's quite specific work. You need proper eyes and actually patience to do that, okay? So, and a bug that maybe disappear after a few crosses. So 10,000 crosses is a lot of time that he spent. Obviously, he was a priest, so he had the time, so he could actually do that. And yeah, that was the main part of his work. As I say, Darwin had a copy, but we don't know if he actually read it. Because if he will have read it, he will actually link what Mendel called elements of inheritance, that we know now they are genes, to his evolution theories. And it will have been really, really important for him, but we don't know if actually he actually had it on his library then. So as I said, he basically discussed there were discrete units or elements of inheritance. Obviously, the war gene was not born yet, okay? So genetics was not born then because there was no a name for it. But there were basically the characters that they will actually give the color of the piece, of the surface of the piece. And that was, for him, quite important to actually resume in the law of segregation and the law of independent assortment, depending if a character is dominant or recessive, okay? And as I said, he was very lucky because he chose two characters that there were no link. They were really far away from the chromosome or in different chromosomes, and actually the independent assortment worked quite well. And we will see more about that later. So Mendel's article was published in 1865. His work was rediscovered, as I say, 35 years afterwards. And then the first time that someone used the word genetics, it was William Bateson, that basically wanted to have a specific science that it was studying inheritance, okay? And that was used from anyone from Greece here? Is that correct? Okay. So I have 50-50, okay? 50% of the students tell me not, and the other 50 are okay. But yeah, so basically it's coming from a work in Greek that basically means origin, okay? So this is basically inheritance. But this was in 1905. Quite often, when scientists have these brilliant ideas, they start to do international conference to sell the new word and to publicize all these things. So as you can see, there were, in the third international conference, it still was not called genetics. It was plant hybridization. And basically, he starts selling it that they should start calling these conferences as genetics to make sure that universities will start teaching that subject, that it was not just part of another subject like cell biology or biochemistry. And it was quite important for them to actually publicize it. The other guy, so Bateson is this guy, Wilhelm Johansen, was the one who actually used for the first time the word gene. So if Bateson called it genetics, the inheritance elements should have been called genes. And that was the first time that they are all described. So in a way, in this moment, they were thinking this is very important and we need to reinvent ourselves and actually put names into that. Just before genetics was born properly, in 1869, Friedrich Mischler discovered the nuclein, that it was a nucleic acid from white blood cells, so basically from pus. He actually extracted DNA. In 1880-1890, Walter Fleming, Strasburger, and Van Beneden, basically independently, they realized that the chromosomes could actually contain the inheritance units that they were talking about with Mendel. And somehow the segregation during cell division and also during meiosis kind of explain the different outcomes of inheritance. So they were starting quite strongly at that time to realize that something was linked there. It was Walter Sutton and Theodore Boveri that basically put the hypothesis on the table and saying that the chromosomes were carrying the genetic material. But as you can see, they basically were following other people's work too. So as I said, Bateson coins the term genetics. Something that we will see later on is this bit that it was discovered quite early on too. Chiasma type. So this is chiasma or crossovers that happen during meiosis. And this is what is allowing the recombination and the independent segregation of characters during the production of gametes. Okay? And this is something that we will see in a second to analyze more about that. But it was quite early on when genetics was born. In 1910, Morgan, anyone knows what Thomas Hunt Morgan worked with? Yeah, the fly. So this is Hunt Morgan doing his crosses, again, very important with the flies, okay? In 1913, Alfred Stetterban makes the first genetic map of a chromosome and actually saw how the genes are in that chromosome in a linear manner. So they're actually one after another. So then basically after the first decade after genetics was baptized as genetics, there was not so much actually to do. They were doing quite a lot of research on all that topics. But then quite early on they found out the problem in genetics. It was to analyze all the data that they were producing. It's a way of all the omics these days. They are producing so much data. The next revolution, it has to be how to analyze this data, okay? So in a way, this is what it happened. So Ronald Fisher, for the first time, published a book about statistics for science that they could actually relate it to genetics. And there was quite a lot of, as we will see in the third lecture next week, there was quite a lot of groups saying that genetics was just one character or it was a sum of different characters. So basically we know these days one gene can produce a phenotype or several genes can produce a phenotype too. So it was basically single inheritance or quantitative inheritance. But at that moment, there were schools of knowledge that they were actually discussing between them which one was the correct one. So then in the 30s, Harriet Crichton and Barbara McClinton identified the crossing over, so this chiasma type that we were talking before, but properly, and actually how this allowed the recombination and the proper assortment of chromosomes in maize. In 1933, so anyone recognize the name Barbara McClintock? This is a Nobel Prize. Does anyone know for what?
So didn't they make the different colours corn?
Yeah, well, she didn't do that. She analyzed that. But why is the different color corn?
Because each one is like really very different colour, isn't it? I can't quite remember the details. This would be all out.
So transposables, transposon elements, transposable movable elements. So the poor woman discovered them in the 40s. They said, you're a woman, you're crazy, we're not listening to you. And it took another 30 years to actually be recognized that she was right, and she won the Nobel Prize. Okay? So all the women here, always go for it if you believe it, okay? Because it's the typical case of science, big thing, big discovery, and no one takes it seriously. And she was a brilliant scientist. She actually was nominated for other Nobel Prizes too, so she was really, really good. So back to the 30s, Jan Brachet identified DNA in chromosomes and the RNA in the cytoplasm cells. But even at this time, the DNA was not seen by quite a lot, most of the scientists, as the material that includes the information for inheritance, for the genes, okay? So most of the people thought they were the proteins because that was where the norm and the fashion was in research at these days. So yeah, for a while, I still didn't know what the DNA was actually the material. In the 40s, very important experiments. That is when where actually they decided that the DNA was that important because they, Tatum and Wells and Biddle saw that the genes code for proteins, and that was quite important. But even so, there were some people thinking that the protein were still the main units of inheritance. That it doesn't make any sense these days, but on those days, especially because the most important guys were working in proteins, they could dictate where science was going. That is something that is happening these days too. So in the 40s, there were more discoveries. As I said, 48, there you have your maze with different colors. Barbara McIntosh discovered the transposons and how there will be units in the DNA that they could move and jump from one place to another. That was completely new. Avery McLeod and McCarthy isolated DNA, and they actually proved that in bacteria transformation was the key material that produced the transformation. Chargaff in the 50s, it was quite important because he came out with a rule that no one could understand at that moment. But he realized that the content in the nuclei, any DNA that he analyzed, adenine equals to the amount of thymidine, and cytosine equals to the amount of guanine. So he was not too sure why, but that was quite important, okay? Why was important? Who can tell me, actually? What of these discoveries afterwards use Chargaff's rule? Anyone? Yeah. So basically they use that to actually produce the proper double helix DNA structure. Okay. Messerson and Stahl demonstrated that the DNA is semi-conservative during replication, so that was quite important, using a template to produce basically two different strands. So basically using what this double helix structure is, you could actually come with different ideas of what was important. During the 60s, there was a huge effort to actually crack the genetic code, to actually know which nucleotides, triplets could actually code for different amino acids. So there was quite a lot of work on that by different people. And this is something that I always put because it's important. This was in the 50th anniversary in 2003 from the discovery of the double helix. And basically it's Watson and Crick in the original picture, and 50 years later, the poor things. But one thing that they realized is that they wrote in the original Nature paper is that, well, I read it. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So that was 53. 3 years later, they discovered that the DNA replication was semi-conservative. So what they say, it was the right thing. One thing that they didn't realize is that also could imply how gene transcription works. And they didn't realize in that time. And actually, Francis Crick worked quite a lot on that, and actually saying he was really pissed off with himself because it took him 15 years to realize how important that structure could have been. So, back to our numbers. So, in the 60s, as I said, normally they were trying to see transcription and how DNA goes to RNA and a protein level. In the 70s, a huge discovery, especially for biotechnology, restriction enzymes and how they could be used for cloning in biotechnology. So that's something that is quite important. And basically, is cut and paste our DNA that you probably have done several times during your degree. 72, they managed to get the first sequence of a gene encoding for a bacteriophage. And then you can see in the 70s the trending is going into actually cloning and sequencing genes. Okay? So the technology was quite starting to be able to sequence the DNA. French Anger's land sequence of an entire genome came at the end of the 70s. And then it's in the 80s when they discovered the polymerase chain reaction proper amplification. Before, here is the first time that Mullis discovered basically the Taq polymerase. So you could heat it up at different temperatures and you didn't have to remove the enzyme or add more enzyme by every cycle that you do in the PCR. So basically it's at the beginning of the 80s where the technology started to boom out for genetics again. Collins and Su actually sequenced the first human gene for cystic fibrosis. And Alec Jeffries, who is a professor in Leicester University, discovered the DNA fingerprinting. Anyone? DNA fingerprinting? Yeah? So just think about CSI. CSI won't exist without this man, Alec Jeffries. And also he is a very nice man who always is against to get databases for humans about genetics markers. So, and he told me the story that basically when he discovered this and sent the article to Nature and got it accepted, suddenly someone at the university told him, do you know that you could make a lot of money with this? So then he had to stop the publication to go the week after to actually get some rights for him. And he didn't use any penny for what he did. He actually made all that money to make sure that companies and countries are not taking advantage. So it's a really nice story of how you use your copyright to actually stop companies making profit and using it for bad things. Quite quickly in the 90s, basically GM came through, okay? And this is something we will see in the final lecture next week. And we will talk more about that, but it's in 1993 the first GM crop went into the supermarkets in the States, and that was called Flavor Saver, although the name has nothing to do with what actually the tomatoes do. So, but we will talk more about that in the final lecture. In 1995, actually was the first genome published, and it was of a bacteria, as you can see. And obviously, after that, most of the genomes that started to appear. And one of them, it was Arabidopsis genome. That Arabidopsis is the model organism in plants, okay? That actually came out in the first year where I was doing my PhD. So it was very handy for my PhD. For the first time, I could do things that no one could have done before in the plant. So it was brilliant. But obviously, we got first yeast published, C. elegans published. We got the human genome quite afterwards, the first draft, and the full after 3 years. And also another plant, rice, that we will see too, is another mother organism has been used since then. And obviously mouse, that is the mother organism for mammalian systems, including humans. So just to summarize all this point of history, basically the units of inheritance of genes were discovered. They knew that they were they could have different forms, and that's what is giving you that variability that is on a genetic pool. That's what we call multiple alleles. And these alleles are basically different mutations that they have happened during evolution. So you can have a different sequence for each of the genes. The genes are DNA. That's something that it was found, and now found in the chromosomes. There was quite a lot of one gene, one protein, but these days we know better. And actually we know that alternative splicing can produce different proteins. One gene actually can produce RNAs that can control all the expression of other genes. We have gene families, as we will see, is quite important also for plant breeding. All the similar genes. Also, different genomes during evolution, as they've been changing from one species to a different species. Actually, we got similar genes doing similar things, but with different sequence, and this is what we call orthologs. And we will see more about that, because they are important for plant breeders. So basically, this is an example of early globulin gene. Nothing to do with plants, as you know, but I always explain like this because it's easier for you to understand. So in mouse, we have two hemoglobins parts. So you got the alpha and beta, and they are very similar. They actually evolve from the same sequence, okay? And this is what we call a paralog, that is a homolog in the same species, okay? In other species, the early globin gene or the alpha chain actually evolve very similar, and they have the alpha globin gene in different species, and the different species actually are orthologs of the mouse, that they are homologs but in different species, okay? So is that okay? Yeah? And the beauty of that is that these orthologs, when you put the sequence of different species, so this is Arabidopsis, it has 5 chromosomes. Soybean has 20 chromosomes, papaya 9, cacao 10, grape 19, poplar 19. So the sequence per chromosome, where is in the different chromosomes and how is the collineality in between them is quite important. And that is very important when we are trying to compare the different species and different varieties in the same species, okay? So, we know Mendel gave the rule of segregation, that basically the different chromosomes are independently assorted on the meiotic recombination, the meiotic production of gametes. So this is an example of actually what is happening for 2 different genes. One for the G with dominant and recessive, and one for the Y gene for dominant and recessive. And as you can see, there are 2 options that they can happen, that the big G and the big I go together and produce 2 gametes with that, or the 2 smalls go together. So in this way, you will have gametes producing the big G's and big Y's together and the small one together. But you could have also something like this, where you got the big G's and the small Y's together, segregated together, and actually mixing together, and the same with the small G's and the big Y's, okay? So what is dependent on that? What is the main thing? Anyone? Why do you have these 2 options? No? So these characters are in different chromosomes. Yeah? The dominant and the recessive, so the big and the small, they have to be in the same chromosome, but in chromosome from dad and chromosome from mom. Yeah? So what is the process that brings them together? Which cells actually produce gametes? What is the process? Yeah. So meiosis is very important for this, okay? And I will explain now in a second what for. So I don't want to bore you too much. I'm specialized in meiosis, so for me it's very important. But we know very little, okay? So what we know for sure is that in early stages that we call prophase 1, the chromosomes, here I painted just one pair of chromosomes, okay? The blue is your dad's chromosome, and the red is your mom's chromosome. As you know, we are diploid species, okay? So that's why it's important to reduce the number of chromosomes to half, to produce gametes with half your chromosome number, so you don't double the chromosome number on your offspring. So what happens during these specific stages is that the two homologous chromosomes, so chromosome 1 from mom, chromosome 1 from dad, are coming together, okay? And there are quite a lot of different processes. I'm not going to go too much in detail because it's not necessary for this. But basically, these chromosomes comes together. You have what we call pairing, synapses, and recombination going on at that time. Recombination means that you are going to have an exchange in between these two chromosomes, an exchange of the DNA sequence in between these two guys. And that exchange is going to allow the most important thing in meiosis, chiasmata or crossover, okay? This is going to allow the two homologous chromosomes, chromosome from mom and chromosome from dad, to be together and go into the spindle in metaphase 1. They are going to co-orientate, and they will be able to separate chromosome 1 from mom to one pole and chromosome 1 from dad to another one. And this is the key part. Basically, you are going to self-link chromosomes, okay? So we don't have just one pair of chromosomes. We have much more than that, and the same for plants, okay? So the beauty of this is that you are going to be able to have the chromosome number, but with the correct information. You will always have a chromosome 1, a chromosome 2, a chromosome 3, and a chromosome 5, okay? But chromosome 1 could be from mom or dad, chromosome 2 could be from mom or dad, and chromosome 3, etc., etc. So that suffering of information is very important, and that's why you are very different from your siblings, okay? And that what it gives variability during meiosis. The other part that is important is once they separate, it's easy to see or to actually measure how much exchange has been in between one of the chromosomes and the others. And that actually we will see too later on that exchange is quite important for plant breeding, because this is when we want to transfer gene information to one to another, or part of the genes or part of chromosomes. As I said, you got half of the chromosomes to one pole, half to the other. And then the second division of meiosis is quite similar to a mitotic division where the sister chromatids just separate, okay? And at the end, you end up having 4 haploid gametes. So this is a process that's highly conserved for unicellular organisms like yeast, highly conserved with mammalian systems like humans, and highly conserved in plants, okay? So we are studying things that they are very similar. They have some differences in the molecular environment, but most of the genes and proteins that are involved in this are highly conserved. So these are the chiasmata, the places where there is actually link and exchange of information between the two homologs. And this is what actually produce these recombinant chromosomes at the end. They are the ones that they have one piece of one chromosome and the other piece of another. Yeah? And these recombinants are what we use, or you probably remember that you use for genetic mapping and genetic problems, okay? So if we come back to the flies and Drosophila and see how the different linkage and map distance were calculated, we're using different phenotypes, different variability and crosses, and then see how they were segregating. Yeah? So that's basically the main thing. You need variability, you select, you cross, pass through meiosis, you have recombination, and you can actually analyze the outcome. So it's basically pure genetics. And in plants, it's the same, okay? Anyone, any questions for this? Okay. So, the genetic analysis, that is the whole thing that we are teaching in this subject, in this module, is basically using that selection to identify different phenotypes that they could be linked to different genotypes, and basically analyze with different biological problems what these genes are doing and why they are important, how they are regulated, all these things. So for this kind of research, model organisms are very important, okay? And these are a few examples of them. So anyone for this one? Yeah, Saccharomyces cerevisiae, yeast. So it's unicellular organism, but a higher eukaryote, and it has been used for lots of genetic and biochemistry research. This one? C. elegans, the worm. Also used as a model organism for humans, these two actually, that they couldn't be more different from humans. But also at molecular level, there are quite a lot of differences on a genetic levels. Okay, so although they select to you, there are good models for disease and things like that. Actually, Drosophila could be better than these two guys. This is Arabidopsis thaliana, that is basically a weed that grows everywhere, but it grows really fast and is completely sequenced. We can do lots of biotechnology techniques, so it's brilliant to work in the lab because basically, as we will see, it requires only 1 month since you put the seeds to collect new seeds. So you can go through quite a lot of generations. Not as fast as yeast or as easy as the worm, but it's a good model actually. Then you got mammalian systems like mouse, rat, that they are fully sequenced. You got lots of different mutants available, antibodies, anything that you can do. And then I put human, although you cannot use it as a model because it's not ethical. But quite a lot of research is done in cell culture. And for genetics, it's a valid system to work. So what model organisms have to be? What are the characters that you need to actually do that? So as I said, a short life cycle is very important because if you have to go through different generations to find an answer, you don't want to be growing something that takes 100 years, okay? Techniques available that you can do genetic manipulation. That's important if you want to produce mutations, if you want to insert different gene copies in it. As we will see, is highly important. No specialist living requirements. This is also important. You don't want to grow something that is really expensive to grow, okay? And very difficult. You want something that grows very, very quickly, very nicely, and without too much to do. That's why Arabidopsis is quite good, because as I say, it's a weed, grows everywhere. So it doesn't require too much. That it has a small genome, and it's easy to sequence. These days, actually, this is changing. Although it was a true fact 20 years ago, these days, because of new techniques of genome sequencing, and the cost is quite low, you can start doing research in more complicated genome sequences, organisms. And so, yeah, one of the things that in plants seems to be very important, on other organisms is not, but in plants it has to have some conservation mechanisms and potential to economic benefits. So that means that the species you are working in, it has some other species, related species, that might be crops that you could actually do something with that. And we will see some examples with that. So in plants, probably the most important, the one thing that is not plant is a moss. That is how basically plants are starting to adapt, or cells start to adapt to actually conquer the land, is Fiscomitella patens. Then two plants, one monocot, rice, and one dicot, Arabidopsis thaliana. So basically, just two different branches of spermatophyta that they came from the vascular plants development. Arabidopsis, so rice, you can see where this economic importance could come from, yeah? Also because it's a cereal, it's not only rice, it's quite similar to other cereals like wheat, barley. Arabidopsis, anyone could link it to anything that it could be?
It's brassica, so that's like cabbage, kale, broccoli, mustard.
There you are. So all the brassicas are very important in production. So this is Arabidopsis, has a very small genome, very clear mutant phenotypes, sometimes, depending on which are you looking at. Rapid life cycle, as I said, in a month you got seeds, basically. It grows and propagates in small spaces. This is how small they are. And there's a huge international community of resources. And these are the brassicas, as you were saying. So these are basically, as I call them, the cousins from Arabidopsis in evolution. So around 80% of the protein level is identical to Arabidopsis. So if you go to the supermarket on your bag, you will have some of these things or products coming from them. So Arabidopsis has 5 chromosomes. As a diploid species, 2n equals 10 chromosomes. So it will have 2 chromosomes 1, 2 chromosomes 2, and so on. So there are really nice genetic maps done by crosses, and analysis of the crosses, physical maps, and the full sequence, basically. So it contains around 25,000 genes, more or less like any other higher eukaryote. All the data is available in internet. You got lots of sequence data, expression data, mutants data. You got, this is an example of the Salk Institute, that they can sell cDNA copies, clones. They can sell different mutant lines for different genes that you are interested. So they have lots of different ways to actually do research on these things, so it's quite good. As I said, it's quite good for collineality, because with species that they are related, you can see where these genes that they are important in Arabidopsis, it will be in other crops, and actually see how they are related, if they are different or not, and actually build up a massive map like this. So it's quite interesting. So I do a break. Yeah? 5-10 minutes? What do you think? 5?